Now, what is, as the name indicates, organogenesis means organ are formed in vitro condition, means organ, and genesis means formation. Organs are formed in the in vitro condition. So we can say that if we talk about the definition, it's the process and initiation and development of organs. It is the initiation. First, we have to go for initiation of formation of organs under in vitro conditions. We can form roots, we can form shoots, we can form leaves from a simple explant. As you can see here, organogenesis is the process where we are able to form shoots, or we are able to form roots, then from root, then to shoot, then again to the whole plant. Or the shoot formation takes place first, then the root formation, then again the whole plant. So this technique was first developed by two different scientists, or they were the first two scientists who reported that formation of shoot and formation of root. White in 1939 was the one who used tobacco hybrid plant, where he was able to go for regeneration of shoot, where he formed the shoot organ first, and he used his explant as a tobacco. And root formation was observed by the Nobet Code in 1931. He takes a root carrot callus, where he observed the root formation. These two scientists were the first who observed root and shoot formation. Shoot formation was observed in tobacco plant, and the root formation was observed in the carrot callus. Now, what are the different processes or events which are occurring in the organogenesis? We all know that whenever we have to start the plant tissue culture, we have to take the explant. It can be explant from any source, plants, leaves, shoot. Then the first step to this explant happens to the differentiation process. When the cells, we have to go for differentiation. The cells which are differentiated will go the differentiation process. It means they will form a simpler cells, or we can call them as a meristematic cells. Then from the differentiation process, they will enter into a redifferentiation process, means they will again differentiate themselves according to the nutrient and the conditions available to them. Sometimes what is happening, that the explant directly enters into the redifferentiation process by the process of the induction and the determination of the cell. First, explant has to induce themselves, that is, they have to get ready for the cell division. Then with cell division, they have to ready, it means induction happen. Then for the determination, that is our redifferentiation, then organs are formed. We are having two different types of organogenesis. First is direct organogenesis. Second is indirect organogenesis. First is direct organogenesis. Second is indirect organogenesis. We all know, first we will be discussing the hormonal combination for organogenesis. Whenever we have to go for induction of shoot, when we have to go for induction or the formation of shoot under in vitro condition, that time we have to take higher concentration of auxin to the higher concentration of cytokinin to the auxin. That is, when जब भी हमें shoot बनाना है ना under in vitro condition, that time we have to take higher concentration of auxin, higher concentration of cytokinin as compared to the auxin. जब भी हमें shoot बनाना है, cytokinin ज़्यादा लेना है, but we have to take both the hormones, but the concentration of cytokinin for shoot formation should be more as compared to the auxin. And when we have to induce root formation, that time we have to take higher concentration of auxin as compared to the cytokinin. As is shown here, we can take 10 times more auxin than the cytokinin, or 100 times more auxin than the cytokinin. Now, we are, as I said, we are having two different types of organogenesis. We can go for direct organogenesis or the indirect organogenesis. As you can see here, we are having both. It's a simple diagram where we are able to, where we will be able to understand how direct and indirect organogenesis takes place. First, we have to take the explant. Here is our explant. We have to take the explant. Like from the plant, we have to take the explant, surface sterilization. All the techniques of the plant tissue culture has to be carried aseptically. We have discussed this. So under aseptic conditions, we are then inoculating our explant onto the slant. Then the merismoids are formed. Then they will start division. Then from directly division, they will not go into the callus formation. They will directly form the leaf primoria or the root primoria. Depends on the culture condition, whether you have provided more oxygen. If more oxygen is there, then the root formation will take place, or if more cytokinin is there, then the shoot formation will take place. This is how the direct organogenesis takes place. Now the indirect organogenesis. In case of indirect organogenesis, we again have to take the explant from the plant source. Then this explant is getting converted, or we form the first callus. Now, if we want to form the callus first, what we have to do, we have to take equal concentration of oxygen to cytokinin. Then only we will be able to get the callus. When callus start dividing, they form the meristemoids, that's meristem cells. Then from the meristemoids, we will get the primoria, root primoria or the shoot primoria. It depends on the medium which you have taken for indirect organogenesis. Now we are having certain factors which are going to affect our organogenesis process. We have to make sure that explant which we are going to take is best in its quality, as its genotype should be good, or it should be able to serve in an in vitro condition. It should be able to serve in an in vitro conditions. Like we have seen in case of, we know that if we take the explant from the woody plants, that's not able to regenerate. Sometimes the regeneration process fails there. So we have to make sure that the explant which we have selected will serve as a tissue source for our organogenesis process. Now, the second is we should be able to know what is the physiological condition or its ontogenic age. It should be young enough. It should be able to compromise all the conditions which are provided in the lab conditions, and in which season we have taken. So we have to take explant in a growing season only, because that time the meristematic cells are very active. They will be able to give best organogenesis result under in vitro conditions. Even the size of the explant matters. Or all the quality of the explant has or play important role in the organogenesis. Now, what culture conditions we have to think when we have to go for the organogenesis? When we are talking about the organogenesis, we should be able to know, we should be able to know that what medium conditions we have to take, whether we have to take the liquid medium or the solid medium. If we have to take the liquid medium, then there should be absence of agar, that is our gelling agent, which solidifies our liquid into the solid medium. A pH plays important role. Mostly in case of organogenesis, we use MS medium, morastic and school media, but the pH we have to keep at 5.8. We have to check the light quality and quantity. It depends on the which plant you are taking, what is the photoperiodism of that particular plant. That photoperiodism you have to maintain under in vitro conditions. Again, the temperature, lead to humidity and gaseous exchange should happen within in the vessel. So there is always cotton plug on the top of your vessel, like if we are taking the slant, or if we are taking the glass vessels like flask. So through these cotton plugs, we should be able to, or there should be enough gaseous exchange. There should be enough gaseous exchange.